based on. It's also to be a pleasure to be here discussing this subject with Gabor. I think we haven't really disagreed so far, so maybe we'll see what will come out from this. And so my task here today are to defend or to show some evidence that tauopathies are a spectrum. And this is my financial disclosures. As Professor Jellinger just mentioned, tauopathies are a broad group of neurodegenerative diseases, and all of them, they have this common feature. They have deposits of phospho-tau protein in the brain. And they can manifest in different ways, either as dementia or movement disorder, a mixture of both, behavioral changes. It can be sporadic, it can be familial. We know about the tau mutation that give rise to some autosomic dominant mutations, but there are also some other variants that cause disease. And as the year passes, we have more tauopathies in our repertoire. So we are always learning more about it. Gabor is leading an international initiative that will finally classify glial tauopathies. I think it's still under-recognized. So we are still learning a lot in this field. But before we really going through tauopathies, I think it's worth mentioning a little bit about the tau gene, that it's very interesting. It's located on chromosome 17. It has 16 exons. And by alternative splicing in human adults, we have 6 isoforms of this tau protein. And this isoform depends on how exon 2, 3, and 10 are part of the isoform or not. So the 3 isoforms that we have, exon 10 or derived from exon 10, we call 4 repeat, and the other 3 we call 3 repeats. We hear a lot about it. So again, in human adults, we have the 6 isoforms. This is different from the fetal humans that we just have 1 isoform. And we have a balance of this 3 repeat with 4 repeat in normal individuals. And the tau protein, it's very important for stabilizing microtubules, and it's present mainly in neurons, but we also find in astroglia, in oligodendrocyte, and one of the main functions is really to stabilize microtubules. So this is a scheme just to show that in normal neurons, usually we find tau in axons. The tau is stabilized in the microtubules. But when you have a disease, the tau goes to different compartments, like the neuronal body, the dendrites, also some in the axons. We find in different flavors. And again, the number of tauopathies are growing over the years. And this is just a representation of some of them. So we have the group of tauopathies that are part of the spectrum of the frontotemporal lobar degeneration. And within this group, we have peak disease, that is a main representative of what we call 3 repeat tauopathies, although we have cases that have a combined glia tauopathy that's actually 4 repeat, and then we have the 4 repeat tauopathies like corticobasal degeneration, progressive supranuclear palsy, and we have globular glial tauopathy. We have AGD, that is a question, is really a disease or not, and we have the familial case. Alzheimer's disease also has a very important component with phospho-tau inclusions, also chronic traumatic encephalopathy, Parkinson complex dementia of Guam, the glial tauopathies, and the list is just growing. So why are they classifying different boxes here? Because as Professor Jellinger mentioned, as pathologists, we can use modern tools like immunohistochemistry, and based on the pattern of the tau protein that we find, the type of cells that are affected and the distribution, it fits some criteria that we can fill the diseases in these boxes. And it works in most of the cases. Some cases there is a gray zone, so especially in the 4R tauopathies, they can have some overlapping. But I think we pretty much can distinguish these tauopathies. So in this sense, these tauopathies are very distinctive. However, if we think about treatment, maybe the story could be a little bit different, maybe because they share some commonalities that I will show here a little further. Maybe we will be able to say that they are also a spectrum of the disease. I just brought some examples of what I was saying until now. So this is an example of progressive supranuclear palsy. In all the cases, I will show we use the same antibody. It's a pan antibody for phospho-tau. It's called CP13. To look at the changes. And PSP is pretty much a subcortical and cortical disease. And here we see the cortex, and here's the white matter. And you can see everything in brown means tau inclusions. And we see that the white matter, it's spare, relatively spare, as opposed to the gray matter. And when we look at the type of inclusions, we can see these globular inclusions in neurons, but PSP has many types of glial inclusions, including this stuffed astrocyte. CDB, cortical-basal degeneration, that is also a 4-repeat tauopathy, it has a completely different aspect. As you can see here, this is again a cortex, this is the gray matter and the white matter. And as you can see here, the white matter is not spare at all in this kind of disease. Actually, the white matter is pretty much affected in CBD. This reflects also in the imaging testing. And what we see is also a mixture of neuronal and glial changes. This is a balloon cell. We hear it a lot, not always we see. We have some astrocytic plaques. They resemble more or less what we see in Alzheimer's disease, but they don't have amyloid. This is a different kind of tauopathy. It's a mixture of 3R and 4R. This is what we see in repetitive brain traumatic injury. And it's called chronic traumatic encephalopathy. It was described around 5 years ago, and now we are hearing much more because as we learn how to identify this, as pathologists, we can really classify these cases. And it has this tau, especially in the deep of the sulcus, around the vessels, and a lot of glial changes, especially in the surface of the cortex. And so again, we talk a lot about tauopathies, how we can distinguish them, but again, is it possible to say that although they are distinctive in the way they are distributed in the brain or the kind of cells they affect, are there common mechanisms that allow us to call them a spectrum? Well, actually there are. So all tauopathies, they have tau hyperphosphorylation. So phosphorylation is normal in tau. It's a way to regulate the protein. But in disease, they become hyperphosphorylated. And then we have this insoluble form, that this is what we detect with these antibodies. And again, they come in different flavors. I showed before peak disease, PSP, CBD. This is just an example to show how they are diverse. But all of them have tau hyperphosphorylation. And tau hyperphosphorylation is not something that we really find in human brains, even in very elderly individuals. Besides the hyperphosphorylation, the majority of the tauopathies, they also have ubiquitin changes, which is a marker for the type of cell death mechanism these cells will undergo. Actually, ubiquitin was also used as a marker before we have good antibodies for specific markers in neurodegenerative disease, so we could identify the disease. And also, there are other changes that we find in all these tauopathies. I would like to comment about tau acetylation. And the findings about tau acetylation came from the conclusion that hyperphosphorylation alone was not enough to cause tau aggregation and toxicity. And so other post-translational modifications were studied. And in 2010, the group of Li Gang from Gladstone in San Francisco, they could show that tau acetylation is not only elevated in human AD brains and rodent models, but in addition, it was required to cause the tau to aggregate and become toxic. So there are some drugs that we can minimize the acetylation, and then we don't see the same toxicity we see when we have acetylation. So we decided to do a study to see how this tau acetylation is really represented in human tauopathies, and we published this about 1 year ago. We used this antibody produced by Li that is against acetylation in the epitope at position 280. And we could show that all in 9 tauopathies that we probe, all of them we could find acetylation, and it was very conspicuous, but not in argyrophilic grain disease that already it's classified as a tauopathy, but it's not necessarily harmful, and we'll talk about it tomorrow afternoon if you are interested. But all the other tauopathies, familial and sporadic, they did have tau acetylation. So probably if this mechanism, if tau acetylation is really so important, we are still studying this, drugs to modulate this probably will be beneficial to all these tauopathies. And finally, I would like to show just a little bit about the familial mutations on the MAPT gene. So there are several mutations that have been identified to date, and most of them cause disease. And besides, despite their mutations in the same gene, in the tau gene, the morphology, isoform composition, and distribution of tau filaments varies a lot. So again, it's a mutation in the same gene that a completely different clinical pathological picture. So suggesting again that it's a spectrum of the same problem. And just to illustrate this is a paper published in 2002 by Lantos and Carnes, and they show in this specific tau mutation the huge variety of tau inclusions that we can find that mimic, for instance, peak disease or this oligodendrocyte that we see in other tauopathies. So these familial cases tell a lot to us. They are rare, but they tell how these diseases can be very variable, even in the same family and in the same kind of mutation. And the last thing I would like to show to corroborate my opinion that tau disease is a spectrum. This is a work that I collaborated in the group with Mark Diamond, who is now in Texas. So he did something very elegant. He took tau fibrils, and he exposed these fibrils to cells, to human cells that had inserted a tau mutation in the binding domain. And when he put these fibrils together with the cells started to produce tau inclusions. But even in the same group of cells that had the same mutation, these tau inclusions were completely different. They could separate, and what they call clones that they interpret as different strains caused by the same artificial fibrils. And these clones, when they were separated and put in petri dishes, and the daughter cells will always show the same kind of distinctive patterns from these strains. This is just a representation. These are kind of puncta, and these are more, these are kind of different dots. So we decided to reproduce the same thing, putting these cells, challenging these cells with homogenates of human tauopathies. We use Alzheimer's disease, PSP, CBD, and AGD. And indeed, what we could see, we use different patients, is that for these diseases, we also got different strains or patterns, and we call this mosaic or order and disorder and so on. And when we put all of this together, we noticed that for Alzheimer's disease, there was a lot of, it was very homogeneous, the kind of changes we found in the cells. But in some other diseases, it was more variable. It might be because, you know, the way we classify neuropathologically, pull together different diseases that look alike, but molecular they are not really alike. But certainly it shows that even for the same kind of this disease, they are distinctive neuropathologically, we can get different kind of seeding in the cells. So I hope I could show you some evidence that tauopathies are a spectrum. And I would like to thank you and to thank my collaborators and people in my lab. Thank you very much.